I told you at the beginning of the last lecture, two different systems of internal communication have evolved in animals. Systems specialized for the transmission of physiological information necessary to coordinate and maintain the activities of the many diverse parts of a complex organism's body. The endocrine system, which we talked about last time, uses chemicals called hormones as messengers that are transmitted throughout the body by the circulatory system. The nervous system, which we'll begin to discuss today, is a very different kind of signaling system, one that uses the transmission of electrical signals along what are essentially specialized cellular wires to send information from one place to another specific place in the body. Now, one major functional difference between the endocrine system and the nervous system as a way to send physiological systems is the relative speeds at which they can send this information. As I mentioned last time, hormonal communication is considered to be a relatively slower way to transmit information, whereas the nervous system is specialized for speed. Hormones can work quickly in some cases. Let's think about that epinephrine example one more time. If I see the flashing light of a police car behind me on the highway, or I'm seriously startled in some other way, I experience what we call the fight or flight response mediated by epinephrine very quickly, maybe only a second or two later. But even a second or two is much too slow for some of the kinds of information that has to be transmitted among the various parts of a complex organism. Imagine, for example, that you're watching someone juggling 3 balls. This little bit of entertainment would well illustrate the need for information transmission on the high end of things. What's necessary for this kind of action to occur? Well, clearly there's information that's traveling between the various parts of the juggler's body. And clearly it must be traveling very rapidly. A number of receptors, which include the juggler's eyes and receptors in her muscles or tendons, are gathering information about the relative positions of the balls and the positions of her hands. This information is being processed and integrated by various parts of a very complex integrator, which is the central nervous system of the juggler, including parts of her spinal cord and brainstem and cerebral cortex. And finally, the central nervous system is sending a constant stream of information out to the many effectors, the muscles that are responsible for controlling the position of the juggler's arms and hands. The nervous system is the mechanism responsible for such rapid transmission and processing of physiological information in animals. And the specialized cells of the nervous system that do this are called nerve cells or neurons. Now, I want to begin this lecture by asking what's special about neurons, these cells that are specialized to transmit electrical information, as compared to other cells. They must be quite different. This is really quite a trick that they're performing. Well, interestingly, the answer to that question, what's different about neurons, is really not much. In most respects, neurons behave like most of the other cells in the body. Now, you might guess that something that's special about neurons must have to do with their electrical properties. Specifically, that there can be what we're going to explain in a second as a voltage difference across the inside and the outside of the cell, across the cell membrane. But such electrical properties are not unique to neurons. In fact, almost all cells in your body have what we call an electric potential difference or a voltage difference across their cell membranes, similar to those observed in neurons. However, there are 3 special things about neurons, and these 3 special things really help outline what we need to understand how neurons function. The first thing is that the voltage across their cell membrane, which I'll explain in a minute, isn't constant, but can change rapidly. In fact, it goes through a very specific kind of rapid change in voltage that we'll later call an action potential. The second special thing about neurons is that this action potential can propagate across long distances. And the third thing is that one neuron can change the voltage in another neuron. So we have changes in voltage, the ability of these voltage changes to propagate long distances, and the ability of one neuron's voltage change to affect another neuron's voltage. Now, the first and third of these specializations, the ability to rapidly change the electric properties of across the cell membrane, and also to change the electrical properties or electrical potential of other cells, can be attributed specifically to specializations, molecular specializations of the membrane itself. Specifically, these properties are due to a kind of protein that we'll call ion channels. Now, the second of these specializations, the ability of neurons to propagate a change in voltage over a long distance, is largely a matter of cellular anatomy, the peculiar shape of neurons. And that's the subject I want to turn to first. So let me begin by describing for you the anatomy of a typical generic neuron. Now, the most obvious difference about the shape of neurons and the shape of other cells is that neurons tend to have several very long processes, long and thin processes, extending away from the body of the cell. Most often, one of these processes, which we'll call the axon, is much, much, much longer than the others, and it may be extremely long. In a giraffe, for example, a neuron that extends from the spinal column to muscles in the leg may be more than 2 meters long. Now, a typical eukaryotic cell body is only about 50 or 100 microns in diameter. So we have a process extending over 2 meters from this very tiny cell. Now, in addition to this elongated axon, there are usually a number of other shorter processes that extend from the body. These shorter processes we'll call dendrites. This is derived from the Greek root dendron for tree because these dendrites often have a branching appearance. Now, the neuron I just described is really a generic neuron. There are neurons that look like this, specifically the neurons that send signals from our brain out to our muscles. But one of the interesting things about neurons is that they actually come in a variety of different shapes. These different shapes largely have to do with the nature of the processes that extend from them, and they also have to do with the particular function that neuron has in terms of transmitting information, the particular geometry across which it needs to gather or send information. So that suggests that these processes have something to do with the transmission of information. Now, this very long extension, we call the axon, at the end of it has another set of structures which we'll call the synaptic terminal. Just to tell you where we're going to call this the synaptic terminal because it will make a connection, or what we'll call a synapse, with another neuron. The synaptic terminal will actually be very closely attached to or aligned with the cell body or the dendrites of other neurons. Now, this anatomy suggests that neurons transmit information from one another along these long cellular wires. And that therefore suggests that the specialization of neurons to be able to transmit information along distances is largely a matter of cell structure, the cells having evolved these elongated processes. Now, I point this out because it contrasts very directly with the way that the endocrine system sends information. Remember, when we talked about the endocrine system, we talked about how information was broadcast, much like radio waves. And the cells that could receive that broadcast were ones that had specific receptors. The nervous system is more like cable TV. An actual wire goes from the source to the end point, and it's along this particular cellular wire that the information travels. So the anatomy of neurons is what enables the long-distance propagation of electrical signals, and we're going to come back to that point a little bit more later on. But first I want to talk a little bit about what these electrical signals really are. All living cells have an excess of electrical charge on the inside relative to the outside. As a result, there's an electrical potential difference, that is a voltage difference across the membrane. Now, to explain that, let me do just a little mental experiment. Let's take what we will call a voltmeter. This is a device that can measure the difference in voltage between 2 points. So this voltmeter would have two what we'll call electrodes, and it will measure the voltage across these 2 points. Now, first, let me just tell you what you would see if you use this voltmeter to measure the voltage across a cell, and then I'll explain a little bit more of what voltage is. If we put these two electrodes into the fluid surrounding a neuron, just the extracellular fluid, we would measure a voltage difference of 0. Essentially, these two electrodes are grounded out relative to each other, and that's because the extracellular fluid is a salty fluid, and electrical charge can travel freely through it. Now, imagine that I take one of those electrodes and leave it on the outside of the cell, but then carefully impale a neuron with the other electrode, so that the other electrode actually enters the cell body of the neuron. Now the two electrodes are separated by the cell membrane. As soon as the electrode entered the cell, I would see a difference in voltage appear. Whereas I was originally measuring a voltage of 0, no difference, all of a sudden when I impaled the cell, I would now measure a voltage of minus 60 millivolts. Minus 60 by convention being with the negative considered negative, with the inside of the cell considered negative relative to the outside of the cell. Now, for comparison, minus 60 millivolts is about 4% of the voltage difference across the two terminals of a typical 1.5 volt flashlight battery. So that's about the magnitude that we're talking about here. Well, now let me tell you a little bit more about what voltage really is. Voltage is a measure of potential energy, really. It's a measure of potential energy that represents the potential to move charges, to move electrons, for example, or charged ions in an aqueous solution. For example, a battery has a voltage, which is an electrical potential difference between its two terminals. If the positive and negative terminals of a battery are connected by a copper wire, then electrons will flow from the negative to the positive terminal, and this flow of electrons could be used to do some kind of work, like light up a light. The voltage measured across the plasma membrane is physically the same thing, but it arises in a very different way. Well, actually not completely different depending on the nature of the battery, but of course it's a biological voltage. When we measure the voltage across a plasma membrane, that is the cell membrane of a neuron, we refer to this in general as the membrane potential of that cell. The membrane potential of a neuron that is at rest, and by that I mean a neuron that is not doing anything actively, it's just sitting there. We call this the resting membrane potential, or just the resting potential. Now, what we want to do is to ask, where does the resting membrane potential of a neuron come from? In other words, how does this potential arise? This is important to take a look at because it will tell us how that potential can change. And it's the change in this membrane potential that we're going to be interested in. The membrane potential of a cell results from two factors. The first factor is that the composition of charged ions differs between the extracellular fluid and the cytoplasm of the cell. Specifically, there are differences in the concentrations of different kinds of ions. The second factor is that different kinds of charged ions are able to cross the cell membrane to a greater or lesser extent. That is, different kinds of ions have differing, what we'll call, permeabilities. Now, both the cytoplasm of the cell and the extracellular fluid that surrounds it contains a whole variety of positively and negatively charged ions. And I want to just sort of walk you through a few of these to orient you. On the inside of the cell, we have a couple of positive ions that we need to look at, and these we'll call cations as well. One of these positive ions is a potassium ion, and another ion that we can find on the inside of the cell, positive ion, is a sodium ion. It turns out that there's a lot of potassium ion inside the cell, but only a little bit of sodium ion in the cell. Now, if we look outside the cell, we also see potassium and sodium ions, but the relative concentrations, the relative amounts, are reversed. On the outside of the cell, there's a lot of sodium and only a little bit of potassium. On the inside of the cell, there's only a little sodium and a lot of potassium. And the point we'll come back to that in a second. Now, there are also a bunch of negatively charged ions, for example, involved in this system. There are many kinds of anions in the cell which really are large charged organic molecules, such as proteins, amino acids, phosphates, and so forth. These large molecules generally all carry a negative charge. On the outside of the cell, the major negative ion is a chloride ion, although some chloride occurs inside the cell as well. Now, here's the key point, the take-home message of this, that there are a variety of ions and their concentrations differ depending on whether you measure it inside or outside the cell. The difference that will be most important for us to keep in mind is the relative concentrations of these two positive ions, potassium and sodium. And to repeat, potassium is high concentration on the inside of the cell and low on the outside. And for sodium, it's just the reverse. Sodium is low concentration on the inside of the cell, but high concentration on the outside. The significance of that will become clear later on. Now, these concentration differences themselves aren't sufficient to fully explain the membrane potential. we also have to consider how easily these different kinds of ions can cross the cell membrane. And to explain what I mean by this, I need to give you a little more background about how ions can pass through a cell membrane. So I need to talk to you a little bit more about the structure of cell membranes. Now, remember that the cell membrane is a lipid bilayer. This is a double layer of lipid molecules called phospholipids. But the lipid bilayer has embedded in it and attached to it a whole bunch of different proteins. And as we've seen now in a couple of different contexts, these proteins really are doing a lot of work of the function of the cell membrane. Now, the proteins that we're interested in are all transmembrane proteins, meaning that they span the membrane from inside to out. And furthermore, they provide essentially channels or openings that allow ions to pass from one side to the other. You can think of these proteins as really forming little donut holes. They're inserted in the membrane, and there's a hole or a pore in the middle that an ion can pass through. This is the only way that these ions can get in and out of membranes, is by going through these proteins. Now, there are a lot of different so-called ion channel proteins, but what I want to do is to just define some of the general properties we can use to classify them. Now, one important property of ion channel proteins has to do with the kinds of ions that can pass through them. Most of the channels that allow ions to pass from one side to the other of the membrane are highly selective to only allow a particular kind of ion to pass through. In other words, there will be a channel that will only allow sodium ions to pass from one side to the other, but won't allow potassium ions to pass. Now, there are some membrane channels that are non-selective, and these non-selective channels will often let many kinds of ions, or in fact, if they're big enough, all sorts of ions move through. So channels can be usually selective and sometimes non-selective, meaning that channels will be specific as to what ion can move through them. A second defining property of ion channels is how the ions move through them. Some ion channels are what we call passive, meaning that ions just move through those channels down their concentration gradient through diffusion. Let me explain just a little bit what I mean by that. In the absence of any other force, a substance will diffuse from a region where it's in a high concentration to a region of lower concentration. So we refer to this substance moving from where it's at high concentration to where it's at low concentration as the substance moving down its concentration gradient, or diffusing down its concentration gradient. For example, if we consider the ion potassium that we already mentioned, and we had a passive channel that allowed potassium to move through it, we would expect, all other things being equal, that potassium would move from the inside of the cell to the outside. Why? Because potassium has a high concentration inside and a low concentration outside. Now, the passive membrane channels of a neuron are important because the passive membrane channels that are are selected for a particular ion will determine the extent or the rate to which that kind of ion can pass through the cell membrane. Imagine it as sort of like a sieve. These ions can move through the holes of the sieve. The more holes they are, the more ions can move through it. If you only have a few holes, the ions can only move slowly. But if you create more holes that are selected for that ion, they can move more quickly. Now, there are, I'll just mention in passing, although we don't need to talk about these quite yet, there are other ion channels that we refer to as active. By active, I mean that they actually pump ions against their concentration gradient. For example, an active channel that was moving potassium across the membrane might be moving it from the outside to the inside up its concentration gradient. Now, potassium is ordinarily going to be pushed down its concentration gradient, and what this means is that these so-called active channels require energy. They actually have to pump these ions uphill, as it were, up their concentration gradient. We'll talk more about where this energy comes from and how it works later in the fifth part of the course. There's a particularly important ion pump, though, that I want to mention at this point, and it's actually a pump that pumps both sodium and potassium, and so it's called the sodium-potassium pump. This is an active ion channel that pumps sodium ions to the outside of the cell, up their concentration gradient, and also pumps potassium ions into the cell, up their concentration gradients. So it's essentially always acting almost as a bailing mechanism for a ship. These ions are always leaking across the membrane through passive channels, going down their concentration gradient, but the sodium-potassium pump is essentially throwing them back against that concentration gradient. This is important over the long run because this is how these concentration gradients are maintained. You can imagine if we didn't have this pump, over some period of time, these ions would run down their concentration gradients and there would be an equal amount of ions, both kinds of ions, on both sides of the membrane. So it's the sodium-potassium pump which essentially is maintaining these concentration gradients. Now, one last defining property of ion channels that we need to consider at this point is whether the channel is always open or whether the channel can be either open or closed. We refer to ion channels that can sometimes be open and sometimes closed as gated channels. They're called gated channels because essentially they have some sort of gate on them that will either open up or close. There are several mechanisms, or really I should say kinds of stimuli, that will open or close gated ion channels. And we're going to look at these kinds of mechanisms a little bit later on when we run into these channels in our discussion of how neurons work. But you might guess that one of these mechanisms is the binding of some chemical to the channel. Because we've seen this before. If some ligand, that's just a chemical that will bind to the molecule, binds, it can change the shape of the protein. And that change of shape essentially will either be effectively opening or closing the channel. As we'll see later, another kind of mechanism that changes the shape of these channel proteins that are gated is the electrical difference across the membrane itself. As the voltage change, this also can open or close channel gates. Now, the key thing to keep in mind at this point is that the permeability of the cell membrane to any particular ion, that is, how freely an ion can cross the membrane, depends on the number of open channels which allow that kind of ion to pass. The more open channels, the more permeable, we would say, the ion is, and the more of it that can cross the membrane. We'll see how almost everything a neuron does has to do with changes in permeabilities of particular ions. Okay, now with that background, let's get back to the membrane potential and see if we can understand something about how it comes into being in a little more detail. To do so, I want you to think just about potassium ions now and how they would move across a membrane. It turns out that a neuron's membrane is actually fairly permeable to potassium. There are a lot of passive potassium channels in the neuron's membrane, and so potassium can cross the membrane fairly freely. Remember also that there's a strong concentration gradient across the membrane for potassium, with the concentration of potassium being high on the inside and low on the outside. Thus, we would expect that there should actually be quite a net movement of potassium ions out of the cell, running through these channels down their concentration gradient. But it turns out that we also have to keep in mind that potassium is a charged ion. So when potassium moves across the cell membrane, down its concentration gradient, it's also carrying excess positive charge with it. So imagine again, for the sake of argument, that we start with a system that has equal charges on both sides. In other words, the two charges are electrically neutral. As the concentration gradient of potassium pushes it outside the cell, down its concentration gradient, it carries with it positive charge. And because it's carrying with it positive charge, the inside of the cell becomes relatively more negative as compared to the outside. Now, the amount of charge that crosses is very little, actually, because it really is only as though there's a cloud of charge just on either side of the cell membrane. If you looked at the cytoplasm of the cell and the extracellular fluid more broadly, they really are more or less electrically neutral. But if you look just across the membrane, there is a charge difference, and this charge difference is because the potassium is leaving the cell membrane, creating positive charge on the outside. But you see, this creates another force that will affect the way that potassium moves. Because the positive charge that builds up on the outside of the cell membrane will now start repelling the positive charge ions, the potassium ions that are trying to go outside. So we really have two forces that have been created. We have a chemical force that is pushing potassium from the inside to the outside of the cell. But as potassium moves out of the cell, an electrical force becomes established, and this electrical force pushes potassium the other way. How much potassium moves? Well, enough potassium will move out until a sufficiently large electrical force is equal and opposite to the chemical force, so that at an equilibrium point we will have a difference in the a net zero movement of potassium across the cell membrane when the chemical force pushing potassium out of the cell is equal and opposite to the electrical force pushing potassium inside the cell. With a few equations, we could actually calculate exactly what the potential difference across that cell membrane would have to be in order for the equilibrium to be established. And it turns out that it's on the order of about minus 85 millivolts. But remember that what we measured when we poked our electrode in that cell was a little bit more positive than that. It was closer to zero. It was more like minus 60 millivolts. So why don't we actually end up with a potential difference that's as negative as we would expect for potassium to reach its equilibrium? Well, that's because we also have this other positive ion, sodium, to consider. Now remember, sodium's concentration gradient wants to send it inside the cell, and also the electrical gradient wants to send it inside the cell as well. And the reason is that being a positive ion, sodium is going to be pushed into the cell by this electrical force, as well as by its chemical force. So you would think then that what's going to be happening if we look at the dynamics of ions moving across this membrane is that sodium would be rushing like mad in the cell. I mean, it's getting pushed by its chemical concentration gradient and by an electrical force. Why doesn't sodium just rush madly in the cell? The reason has to do with permeability. You see, the cell's membrane doesn't have that many passive channels that are selective for sodium. So the relative amount of forces that will eventually contribute to the overall resting membrane potential of a neuron includes not only the concentration gradients of the ions we're interested in, specifically in this case potassium and sodium, but also what their relative permeabilities are. In a resting membrane, in the membrane of a resting nerve cell, potassium is about 20 times more permeable than is sodium. And what that means is, without doing the math specifically, that potassium dominates the solution, as it were, to the resting membrane potential. Most of what we would expect for the resting membrane potential comes from the forces that are acting on potassium. But it's modulated somewhat by the forces acting on sodium. Sodium is not impermeable. It can cross the membrane, but only about a 20th as much. So that means that the effect of sodium being pushed into the cell, making the cell relatively more positive, causes our equilibrium value for the cell membrane potential to be more positive than we would expect for potassium alone. Instead of minus 85, it gets pushed up closer to zero, in the direction of zero, towards about minus 60 millivolts. Well, to summarize that, the take-home message here is that the resting membrane potential of a neuron, or really of any cell, depends on two factors. First, it depends on the concentration gradients of charged ions that can cross the membrane, but second, and equally importantly, it depends on the relative permeabilities of those ions, how easily they can cross the membrane. Now, why should we be interested in any of this? Well, to answer that question, I want you to imagine for a second what would happen if we played a little trick on this cell membrane and we somehow made sodium more permeable. Remember, potassium is very permeable and sodium isn't. But let's say we could all of a sudden introduce a whole bunch of channels that would allow sodium to rush to cross the cell membrane. What would happen? Sodium would indeed rush into the cell, and if we were measuring the difference across that cell membrane with our electrodes, we'd see that the cell membrane would go from a negative value on the inside, minus 60, to a very positive value. In fact, it would go up to plus 50 or so millivolts, radically changing. This is exactly what happens when a neuron becomes active, or does what we call fires an action potential. And that's the topic we're going to turn to in the next lecture.